Chapter 7, Membrane Structure and Function. In the last chapter, you were taken through a tour of the cell where you were introduced to the organelles and the network of cytoskeleton components that gives organization and structure to the cell. Knowing the organelles allows you to distinguish between prokaryotes and eukaryotes and will help in future physiology courses. You also learned about the selective barriers surrounding every cell called the plasma membrane. In this chapter, we will dive further into the structure and function of the plasma membrane and the implications for cellular functions. Specifically, you will learn how to describe or model the endomembrane system and the cell membrane with respect to secretory and absorptive processes. You will describe osmoregulatory processes at the cellular level, describe cellular level gas exchange by diffusion as supporting organism function, and describe or model some details of the basis of tissue level structure and function. Let's review the structure of the plasma membrane briefly before we continue. Knowing the structure of the membrane is crucial to understanding how it functions. The plasma membrane is the boundary that separates all living cells from its surroundings. Every single cell has a plasma membrane. Plasma membranes are comprised of a special type of lipid called a phospholipid. Recall from Chapter 4 that a phospholipid is two fatty acids and a phosphate group attached to a glycerol. The two fatty acids are hydrophobic hydrocarbons, but the phosphate group forms a hydrophilic head. This makes the phospholipid both hydrophilic and hydrophobic. This type of molecule is called amphipathic. Part of the molecule is water-loving, while part is water-fearing. The phospholipids orient themselves so that the hydrophobic tails are sheltered inside the membrane, while the hydrophilic heads are exposed to water on either side. This orientation gives the membrane selective permeability. Selective permeability means the membrane allows some substances to cross more easily than others. It's very important that you understand this concept before we continue. Most molecules in your body are not able to cross this phospholipid bilayer, which we call the plasma membrane. This is crucial to the cell's ability to regulate its internal environment and maintain a favorable homeostasis. Nearly all of the cells in our body are in contact with the bloodstream, which acts as a highway carrying all nutrients, gases, signaling molecules, and even toxins like alcohol throughout the body. It's necessary for the cell to be able to regulate what can or cannot enter, and the cell membrane is the first and most important barrier of defense in this process. The second layer of defense are called transport proteins, which we will focus on in this chapter. Transport proteins are responsible for controlling passage across cellular membranes. To understand how the plasma membrane functions, you have to understand the structure. Cells are the smallest units of life and must therefore perform all cellular operations required of life. To achieve this successfully, the plasma membrane possesses a unique property of fluidity, whereby the phospholipids and all protein components of the membrane can shift and move through the bilayer. This is termed the fluid mosaic model. The membrane is a mosaic of protein molecules bobbing in a fluid bilayer of phospholipids. However, don't mistake this to mean that the cell has no control of the fluidity or mosaic of proteins. Proteins are not randomly distributed in the membrane. The cell is a finely tuned piece of organic machinery, and everything is highly orchestrated to maintain homeostasis in the support of life. Let's take a look at how the cell monitors the fluidity of the membranes. Recall from Chapter 5 that when phospholipids are dispersed in water, they will self-orient themselves into a bilayer, with the nonpolar hydrophobic tails facing inward and the polar hydrophilic tails outward in contact with the polar water. The main type of bond holding these phospholipids together are weak hydrophobic interactions. Because these bonds are weak, the phospholipids have flexibility to move around within the phospholipid bilayer. Don't forget, the three types of lipid molecules are fats like oils, phospholipids, and steroids. Just like fatty oils such as olive oil are slick and fluid, so too is the phospholipid bilayer. This means that some proteins contained within the bilayer may also move sideways within the membrane. A study investigating membrane fluidity showed that when two cells with different antigens, shown here with pink mouse cell antigens and purple human cell antigens, are hybridized, the antigen proteins will mix within 1 hour. Again, this is due to the fluidity of the phospholipids. Do note that movement is free sideways or laterally within the membrane, but there are rare proteins that can actually flip-flop across the membrane from one phospholipid to another. Scientists were very profound creating the names of these proteins. They're called flipase and flopase. There are a few ways the cell can tinker with the plasma membrane to affect fluidity. This is important for the cell's ability to effectively respond to varying environmental conditions to maintain an optimal physiological environment. The first is temperature. As temperature cools, membranes switch from a fluid state to a solid state. You experience this at the macrocosmic level when you heat butter to melt it. The temperature at which a membrane solidifies depends on the types of lipids. For example, membranes rich in unsaturated fatty acids are more fluid than those rich in saturated fatty acids. Membranes must be fluid to work properly. Membranes are usually about as fluid as salad oil. The last thing affecting fluidity is the steroid cholesterol, which has different effects on the membrane of animals at different temperatures. At warm temperatures, such as 37 degrees Celsius, cholesterol restrains movement of phospholipids. At cool temperatures, it helps to maintain fluidity by preventing tight packing. Though cholesterol is present in plants, they use related steroid lipids to buffer membrane fluidity. Now let's look at membrane proteins and their functions. Somewhat like a tile mosaic, a membrane is a collage of different proteins, often clustered in groups, embedded in the fluid matrix of the lipid bilayer. Phospholipids form the main fabric of the membrane, while proteins determine most of the membrane's functions. There are two major categories of proteins within the plasma membrane: peripheral proteins and integral proteins. Peripheral proteins are bound to the surface of the protein. Periphery means outside the boundary of something. Integral proteins are different. Integral proteins penetrate the hydrophobic core of the membrane. Some integral proteins even span the entire membrane, and these are called transmembrane proteins. Trans means across. This is an image of a transmembrane protein. Notice how it spans across the entire membrane from the extracellular side to the cytoplasmic side. Extracellular means outside the cell, and of course the cytoplasmic side is the cytoplasm within the cell. The hydrophobic regions of an integral protein consist of one or more stretches of non-polar amino acids, often coiled into alpha helixes, just like you see here. This protein has 7 transmembrane domains running through the membrane. Cell surface proteins can carry out several functions, including transport of molecules across the membrane, enzymatic activity to speed up biochemical reactions, signal transduction to catalyze an intercellular response to an extracellular stimulus, cell-cell recognition for immunological responses, intercellular joining for stability and structure, and attachment to the cytoskeleton and the extracellular matrix, or ECM. Cell surface proteins are important in the medical field. In the example shown here, HIV must bind to the immune cell surface protein CD4, shown in purple, and a coreceptor, CCR5, shown in green, in order to infect a cell. HIV cannot enter the cell of resistant individuals who lack CCR5 coreceptor. Drugs are now being developed to mask the CCR5 protein. Carbohydrates can play an important role in cell-cell recognition. Cells are able to recognize each other by binding to molecules, which often contain carbohydrates, on the extracellular surface of the plasma membranes. Membrane carbohydrates may be covalently bonded to lipids, forming glycolipids, or more commonly to proteins, and these are called glycoproteins. Notice the root words here. Glyco means sugar. A glycolipid is a fat with a sugar on it, and a glycoprotein is a protein with a sugar on it. Carbohydrates on the extracellular side of the plasma membrane vary among species, individuals, and even cell types in an individual. So you now know that phospholipids orient themselves into the phospholipid bilayer, forming the plasma membrane. But the plasma membrane isn't the only part of the cell made of phospholipids. All of the organelles in the cell use phospholipid bilayer as their outer membrane. This includes the nucleus, mitochondria, endoplasmic reticulum, the Golgi apparatus, and vesicles used for transport within the cell. In the last chapter, we learned about the functions of each of these organelles. Take a look at the picture showing the flow of material through the cell's organelles. The ability of the ER to bud off into a vesicle, for that vesicle to fuse with the Golgi apparatus, and then for vesicles released from the Golgi to fuse with the plasma membrane for secretion, is crucial to the endomembrane system. The fact that all membranes are made of the same phospholipids allows for easy movement of materials throughout the cell. You'll also notice from this picture that there is a sidedness to the membranes, meaning they have distinct inside and outside faces. Each side has an asymmetrical distribution of proteins specific to that compartment's specialized role in the cell. We've already discussed the structure of the membranes and the resulting selective permeability. A cell must exchange materials with its surroundings, which is controlled by the plasma membrane. The selective permeability allows for tight regulation of molecules trafficking into and out of the cell. So what molecules are able to move freely through the membrane? Hydrophobic, nonpolar molecules, such as hydrocarbons, are able to dissolve in the lipid bilayer and pass through the membrane rapidly. Hydrophilic molecules, including charged ions and polar molecules, do not cross the membrane easily. Remember this phrase, like dissolves like. Large molecules of any type will have an issue crossing the membrane and require another form of transport. So to answer the question, what can move freely through the plasma membrane? Small, nonpolar molecules, like oxygen and carbon dioxide, as well as water. So if only small nonpolar molecules and the polar water molecule can pass through the plasma membrane unaided, how do we get everything else into the cell? Transport proteins. Transport proteins allow the passage of hydrophilic substances across that membrane. We have two major types of transport proteins, channel proteins and carrier proteins. Channel proteins have a hydrophilic channel that certain molecules or ions can use as a tunnel. One very specialized type of channel protein is called an aquaporin, which greatly facilitates the passage of water molecules. Carrier proteins bind to molecules and change shape to shuttle them across the membrane. Whether it is a channel or a carrier protein, all transport proteins are specific for the substance that it moves. Before we talk further about the proteins involved in protein transport, we have to learn the concept of diffusion, as this is how much of the transport is propelled. Diffusion is defined as the tendency for molecules to spread out evenly into the available space. At the atomic level, molecules are vibrating and moving due to thermal and kinetic energy, and therefore randomly bumping into one another. Diffusion is therefore random. However, although each molecule moves randomly, diffusion of a population of molecules may be directional. At something called dynamic equilibrium, the same amount of molecules are crossing the membrane in one direction as in the other. Look at this depiction of diffusion. In image A, we see two sides separated by a permeable gray membrane. On one side, we have yellow molecules of dye suspended in water. With time, the molecules can begin to randomly interact and move until eventually they spread evenly. We see for the first two images there is an overall net diffusion to the right. In the top left image, there is only diffusion to the right. In the middle top image, there is diffusion going both ways, but the overall net diffusion is still to the right. The molecules want to spread out, and there are more on the left, pushing the net diffusion to the right. In the image on the top right, we see this solution has reached equilibrium. Equilibrium does not mean equal concentration. Equilibrium means equal rates of diffusion. In other words, just as many yellow molecules are moving to the right as to the left. In image B, we have two different solutes, yellow and purple, separated by a permeable membrane. Again, the molecules randomly interact and diffuse into available space. This is a net diffusion from where the solute is most concentrated to where it is least concentrated. In the final panel, we see equilibrium for both solutes, which is equal rates of diffusion in both directions. Again, not equal concentration. In diffusion, substances like those solute molecules diffuse down their concentration gradient, which is the region along which the density of a chemical substance increases or decreases. In other words, substances diffuse from areas of high concentration to areas of low concentration. No work must be done to move substances down their concentration gradient. So when a substance diffuses across a biological membrane, it is a passive transport because no energy is being expended by the cell to make it happen. In the last example, we saw the diffusion of solutes down their concentration gradient. Water can also diffuse across a selectively permeable membrane. This has a special term called osmosis. Pay very close attention to this explanation. Water diffuses across a membrane from an area of lower solute concentration to a region of higher solute concentration until the solute concentration is equal on both sides. Take a look at the image on the left. This hydraulics tube is separated into two sections by a selectively permeable membrane. In this case, the membrane is only permeable to water and not to the green sugar solute. Water is going to want to diffuse down its own concentration gradient, which means it will move from an area of lower solute concentration to an area of a higher solute concentration. The left and right sides of the tube begin with equal amounts of water, but unequal solute concentrations. The left is less concentrated than the right side. So water will move from the left towards the right, from lower solute concentration to higher solute concentration. The osmotic pressure is so great that the water level on the right exceeds the level on the left. Osmosis created a more similar concentration of solute on both sides. Now that you know the basics of diffusion and osmosis, we can apply this to cells. As I keep mentioning, homeostatic balance is crucial to all aspects of cellular physiology, including the proper balance of water in the cell. Tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. The tonicity of that solution depends on its concentration of solutes, which cannot cross the cell membrane. There are three types of tonicities: isotonic, hypertonic, and hypotonic. Let's start with isotonic as it's the easiest one to understand. Isotonic solutions have a solute concentration that is the same as that inside the cell. Therefore, there's no net movement across the plasma membrane. The central panel shows an isotonic red blood cell, which maintains its proper shape and therefore function. Notice there's water diffusion both into and out of this cell. Osmosis is therefore in equilibrium here, with no net change in movement. If you've ever had a saline drip intravenously, that saline IV would need to be a concentration of salts that are isotonic to your blood. Otherwise, the saline IV would create a net diffusion of water into or out of your cells, which could dehydrate or burst your cells. The animal cell likes the isotonic environment. The plant cell in the middle panel is considered flaccid in an isotonic solution, which is not ideal. The next type of tonicity is hypertonic solution. In a hypertonic solution, the solute concentration is greater than that of the concentration inside the cell, and therefore the cell loses water. The panel on the right shows cells in hypertonic solutions. The solution has a solute concentration greater than that within the cell, and water wants to create equal concentrations. Therefore, water will diffuse from an area of lower solute concentration inside the cell to an area of higher solute concentration outside the cell. This causes the red blood cell to shrivel, a process termed crenation. The plant cell has also shriveled. Notice the difference here though. We learned in the last chapter that plant cells have sturdy cell walls in addition to plasma membranes. The cell walls do not buckle, but the plasma membranes have shriveled and pulled away from the cell walls here. This is called plasmolysis. The last type of tonicity is hypotonic. In a hypotonic solution, the concentration is less than that of inside the cell. Therefore, the cell gains water. The panel on the left shows cells in hypotonic solutions. The concentration of solute in this solution is less than the concentration within the cell. Once again, water wants to move from an area of lower solute concentration to an area of higher solute concentration. Therefore, water rushes into the cell and causes it to swell until it bursts or lyses. The plant cell, on the other hand, is thriving in the hypotonic solution. Water rushes into the cell, but it does not burst because it has the strong cell wall to maintain its structure. This cell is said to be turgid and is the optimal form of plant cells. This is why plant cells love water. Water has far lesser concentrations of salts and nutrients than the actual plant cells, and therefore osmotic pressure will cause the diffusion of water into plant cells and keep them turgid. Now you see how important it is for cells to maintain the proper water concentrations. This process is termed osmoregulation, which is the control of solute concentrations and water balance, a necessary adaptation for life. Some cells that live in extreme conditions have special adaptations to maintain their water balance, such as the bacteria and archaea that live in hypersaline environments. So we have completed a review of diffusion and osmosis. These are both types of transport called passive transport because no energy was used. The movement of molecules in diffusion is driven solely by concentration gradients, differences in concentration across the plasma membrane. Diffusion is also unique in that it uses no transport proteins. It only has to do with movement of water or solutes across a selectively permeable membrane. Now we have to learn the other type of passive transport, called facilitated diffusion. This type of diffusion does use proteins to aid in transport, but still does not require energy. In facilitated diffusion, transport proteins speed the passive movement of molecules across the plasma membrane. Transport proteins are the two types we discussed earlier in the lecture, channel proteins and carrier proteins. Remember, channel proteins provide corridors that allow a specific molecule or ion to cross the membrane. One special type of channel protein is an aquaporin, which facilitates diffusion of water and helps to maintain fluid homeostasis in several tissues, including kidney, lung, GI tract, and the neurons in your brain. Here is a model of an aquaporin transporting water molecules across a membrane. The yellow molecule is a water molecule highlighted to help you follow the flow of the molecules. There are also basic ion channels which facilitate the transport of ions. Some of these ion channels, called gated channels, open and close in response to a specific stimulus. A prime example is in nerve cells during an action potential. Sodium and potassium ion channels open in response to electrical stimulus. You'll learn more about that in Physiology 1 or in your psychology courses. In image A, we see a channel protein. Notice how it is a transmembrane protein, allowing solute particles to move from the extracellular space, where the concentration is highest, to the cytoplasmic intracellular space. This is driven by diffusion and is passive transport. In image B, we see the carrier proteins which undergo a conformational shape change to translocate the solute across the membrane. This change in shape is triggered by the binding and release of the transported molecule. So now you've been introduced to all three types of passive transport: simple diffusion across the plasma membrane, facilitated diffusion with a channel protein, and facilitated diffusion with a carrier protein. Passive transport means no energy is used by the cell to transport these molecules. The molecules simply move down their concentration gradients. Now we'll cover active transport, which does use energy, usually in the form of ATP hydrolysis. Whereas passive transport moved solutes down their concentration gradients, active transport moves solutes against their concentration gradients. In other words, active transport moves solutes from a region of lower concentration to regions of higher concentration. All proteins involved in active transport are carrier proteins. This ability for active transport proteins to move solute up their concentration gradients allows cells to maintain concentration gradients that differ from their surroundings. For example, an animal cell has a much higher potassium and a much lower sodium concentration compared to its surroundings. This is controlled by the sodium-potassium pump, a transport protein that is energized by transfer of a phosphate group from the hydrolysis of ATP. So let's walk through the basic steps of how the sodium-potassium pump functions. The overall goal is to move 3 sodium ions out of the cell and 2 potassium ions in. That's 3 Na out and 2 K in. As we begin, the carrier protein is open facing the interior of the cell. 3 sodium ions are able to bind. In step 2, the energy molecule ATP is hydrolyzed, resulting in the transfer of a phosphate group onto that carrier protein. This activates the carrier protein for movement. The protein then has a conformational shape change and releases the sodium ions to the outside of the cell. In step 4, 2 potassium ions are able to bind and the phosphate group is removed, resulting in the final conformational shape change seen in step 5. In step 6, the potassium ions are released and the process is able to repeat. Each cycle requires the hydrolysis of one ATP molecule. You now have the basics of how ions and molecules are moved across cell membranes. Remember, all of this is tightly orchestrated to maintain homeostasis. Cells need to maintain certain gradients in order to create environments that drive diffusion. One important gradient is the charge gradient, or the difference of positively charged ions versus negatively charged ions. For example, neurons need to maintain an overall charge of negative 70 millivolts. The inside of a neuron is negative in comparison to its surrounding extracellular environment. There's a special term for this called membrane potential, which is the voltage across the membrane. Voltage is just charge and is measured in millivolts for cells. Voltage is created by differences in the distribution of positive and negative ions across a membrane. The cytoplasmic side of the membrane is negative in charge relative to the extracellular side, which is positive. Another important aspect of cellular physiology is something called the electrochemical gradient, which drives the diffusion of ions across the membrane. Two things make up the electrochemical gradient: the chemical force and the electrical force. The chemical force is the ion's concentration gradient. The electrical force is the effect that the membrane potential, governed by difference in charge, has on the ion's movement. Looking at a neuron, the membrane potential is negative 70 millivolts. Again, this means that the inside of the cell is more negative than the outside. There are many negative and positive ions that factor into this overall net negative 70 millivolt charge. Let's look at two ions, sodium and potassium. The inside of a neuron has a higher potassium concentration and a lower sodium concentration than the outside. When gated sodium channels open, the electrochemical gradient favors the diffusion of sodium into the cell, and therefore sodium will rush in. Both the chemical force and the electrical force favors this movement. If gated potassium channels, K plus channels, are opened, the chemical force would favor potassium to rush out, but the electrical force would favor positively charged potassium to rush in. The interplay between these two forces, chemical and electrical, results in a net movement of ions across the membrane. An electrogenic pump is a special transport protein that generates voltage across the membrane. The sodium-potassium pump is the major electrogenic pump of animal cells. The main electrogenic pump of plants, fungi, and bacteria is a proton pump, which actively transport hydrogen ions out of the cell. Electrogenic pumps help store energy that can be used for cellular work. We also have something called co-transport. Active transport of a solute can indirectly drive transport of another substance. This is what we call co-transport. In this process, the diffusion of an actively transported solute down its concentration gradient is coupled with the transport of a second substance against its own concentration gradient. In this example, the proton pump acts as an electrogenic pump by actively pumping protons out of the cell. This creates a voltage across the membrane and was done using energy. H plus can then diffuse down its concentration gradient through the H plus sucrose co-transporter. As it moves down its concentration gradient without any energy, it pulls the sucrose along with it as a co-transporter. The last type of active transport is called bulk transport, which is movement of large molecules such as polysaccharides and proteins across the membrane in bulk via vesicles. This large movement of molecules requires energy. There are two main types of bulk transport: exocytosis and endocytosis. In exocytosis, transport vesicles migrate to the membrane, fuse with it, and release their contents outside the cell. Cells that specialize in secretion rely on exocytosis to export their products. Just see the root of that word, exo and cytosis. Cyto means of the cell, and exo means outward. In endocytosis, the cell takes in macromolecules by forming vesicles from the plasma membrane. Endocytosis is a reversal of exocytosis involving different proteins. There are three types of endocytosis. We have phagocytosis, which is cellular eating, pinocytosis, which is cellular drinking, and receptor-mediated cytosis. And again, if you look at the root word, endocytosis, endo means inward, and cyto means of the cell. Pictured here are the three types of endocytosis. Remember, in endocytosis, the cell is bringing material into the cell. In phagocytosis, which is cellular eating, a cell engulfs solid particles in plasma membrane to form a food vacuole. In pinocytosis, which is cellular drinking, molecules that are dissolved in droplets are taken up when extracellular fluid is gulped into tiny vesicles. And in receptor-mediated endocytosis, binding of a specific solute to receptors that are on the outside of the cell triggers the vesicle formation and triggers the cell to bring that in. Human cells use receptor-mediated endocytosis to take in cholesterol, which is carried in particles called low-density lipoproteins, or LDLs. Individuals with the disease familial hypercholesterolemia have missing or defective LDL receptor proteins. When you're done, take a moment to review and test your knowledge in the questions that follow in the PowerPoint. And in the next chapter, we will move on to an introduction to metabolism.